Okay, so we'll start with neoplasia today, and let's see how much we'll be able to like cover. So before we begin, right, neoplasia is basically before we begin. So neoplasia, as the word says, it's basically disorganized and uncontrolled like cell growth. There's too many cells, and the problem is that these multiple cells they're not working well. Both of these things combined give us the problems that cancer has. So if we were to generalize what problems cancer has, what would we be able to say? Like, what are the symptoms or what are the problems with cancer or neoplasia? Oh, wait. Wait, I wasn't able to hear. Could you repeat that? Yeah, so that is true. You have multiple cell growth. And the problem is that all of these different cells, right, they're trying to steal nutrition from the other healthy tissue. They're also trying to kind of invade into other tissue. So they're causing a mass effect. If there is more pressure on, let's say, a specific organ, then that organ might not start working properly. Additionally, this cancer, it might also travel to other places. It can disrupt multiple different functions, and that is all the problem which we have with cancer. Now, when we talk about neoplasia, neoplasia is a very specific type of cellular adaptation. Initially, we might have something as hyperplasia. After hypoplasia or hypertrophy, we might have some kind of metaplasia. After metaplasia, we have dysplasia, which is a disorganized cell growth, but this is not yet cancer. It's not yet neoplasia. So, do you guys remember what's the difference between dysplasia and neoplasia? Oh, I meant dysplasia. Dysplasia versus neoplasia. Sorry, I accidentally said metaplasia. Dysplasia versus neoplasia. Yeah, but we can say the same thing for cancer. Cancer is also loss of uniformity, loss of orientation of the cell, changes in cell shape, size, function. But then, what would be the difference between these two? Yeah. So Kizzy actually said it correctly that dysplasia is reversible and neoplasia is irreversible. Now, dysplasia it can be reversible, and even though there is a very small percentage that dysplasia would be reversed, it still is reversible. Now, once you come into the category or in the territory of neoplasias, then we know that this is irreversible. You will have to treat it with chemotherapy or surgically remove it. There's no other option. So, there are multiple phases as to how cancer progresses. The first phase is that cancer has developed. This is disorganized cells, they're irreversible, they're all just growing very badly. But this part, you see, they're within the basement membrane. So they've not crossed the. This is called a carcinoma in situ. That means the cancer is developed, but it's not crossed the basement membrane yet. The next type of cancer is when you have a cancer which has invaded the basement membrane, and this is usually like a malignant tumor which can then invade the blood vessel. And once the cancer does invade the blood vessel, at that point we have the metastasis. So this is normal tissue, dysplasia, dysplasia goes to carcinoma in situ, which goes to malignant tumors, and then you can have metastasis. Now, sometimes, these cancers, they don't always have to invade the basement membrane. Sometimes they can be above the basement membrane and constantly grow. Those are benign tumors. And benign tumors are better because they don't metastasize to multiple different places. Oh, actually, there is one more kind of thing that I would say is that whenever you have a mass, whenever the cancer is very well localized, at that time you can think about surgically resecting the cancer. Yeah, because it's all in one place, so you can easily just surgically remove the entire thing and then you can extract it. On the other hand, if you have a cancer which has metastasized, it's gone to multiple different organ systems in the body. At that time, you're thinking about treating with chemotherapy. Why chemotherapy? Because when the patient does take chemotherapy, it will be in the blood, and then through the blood, it will supply all the different organs. So that's also how the treatment approach is different for these cancers. Oh, okay. Yeah. So carcinoma, benign and malignant. Now, based on what type of epithelium we have, the cancer is called something else. So let's say if you have a cancer of blood vessels, it's called a hemangioma. Now, here there is one very important thing. Whenever you hear the word oma, it basically means a mass of something, and this is usually a benign mass. Oma is always benign. On the other hand, if you ever hear the word carcinoma or sarcoma, that suggests that the cancer is malignant in nature. So here you have carcinoma, and then here you have sarcoma. So both of these are considered malignant in nature. Now, here they say that if you have a cancer of the blood cells, that means you have a cancer of, let's say, RBCs, WBCs, maybe even platelets. They are called leukemias or lymphomas, and they're always malignant. So, why do you think these are always malignant? Why are blood cancers always malignant? Yeah, because blood cells are supposed to be in the blood, and if a cancer is in the blood, it's already malignant. And yeah, they'll have to travel in the blood too. Through the lymph node, it's much more easier. So both of those reasons. Now, normally, a lot of cancers, when we say they're malignant, they travel through the lymphatic system. However, there are a few which do travel hematogenously. That means they travel through the blood. And there are four particular cancers. So, do you guys remember the name for all of these four? These are very important to remember. These four cancers which travel in the blood. No? Okay. So, the first one is renal cell carcinoma, which can travel in the blood. The second one is hepatocellular carcinoma, so a liver cancer that can travel in the blood. We have malignant melanoma, which can travel in the blood. And then finally, we have follicular carcinoma of thyroid, which can also travel in the blood. Yeah. So now these are all the other type of cancers, depending on if they're benign or if they're malignant. Can you please repeat once again? The four cancers. Yes. Yep. So these are renal cell carcinoma, hepatocellular carcinoma, malignant melanoma, and then follicular carcinoma of thyroid. But just check once if it's follicular carcinoma or if it's medullary carcinoma of thyroid. It is one thyroid cancer. I think I forgot the name for it. Now, here we also need to differentiate cancer based on its grade and then based on its stage. Now, You can do both of these in real world. We do want to do both of these, but at least for the exam, they'll usually tell you to focus on one of these, and majorly they'll tell you to focus on the grade, the stage of the cancer. So, what does the stage of the cancer mean? It's divided into three components. So, T, N, and M. So, the T means the size of the tumor and how big the tumor is. The N means has it invaded towards a regional lymph node, and the M last is the metastasis. So, that is like has it metastasized to multiple different places. Now, obviously, metastasis is the most important for us to know because it tells us how severe the condition is, how severe the cancer is. If you do not have metastasis, then you can think that okay, maybe the cancer has traveled to the lymph node, and if it's not traveled to the lymph node, then we want to think what is the size of the tumor and how deeply is it embedded in the original tissue. Now, on the other hand, grading it's divided into only two like subcategories. Either you have a low grade tumor or you have a high grade tumor. In low grade tumor, basically it means that these are well differentiated. That means the cancer it looks very similar to the surrounding tissue. On the other hand, a high grade tumor will have cancer cells which look very different from the surrounding tissue. So even here, you can see that in the low grade tumor, we can still see the nucleus, we can still see the edges of the cell, and all of these cells will look similar. On the other hand, for a high grade tumor, we do not see the nucleus. Actually, we can. The bigger, like darker circle is the nucleus, and you see how big the nucleus is here. And all of these cells they look very different from the other cells. So that is a high grade tumor. High grade tumors are much worse compared to low grade tumors. Okay. Now we have these hallmarks for cancer growth, and I wouldn't say these are particularly important because these are like things we've already discussed. So basically, cancer ultimately is a group of cells which have kind of given up on the rules of the body, and they simply want to focus on themselves. So this means that cancer would like to grow ultimately, like infinitely, as long as it's possible by the body, as long as it can be sustained by the body. So the cancer will try to increase growth signals that it gets, and these are done in the like these are done by some genes which are PDGF, maybe some signaling molecules such as RAS, maybe some transcription factors which are MYC, and then cell cycle regulators such as cyclins and cyclin dependent kinases. Now, all of these different molecules they can give rise to specific cancers. So, as an example, the MYC gene, a smaller variant, the N, sorry, not the N, the LMYC gene will give rise to lung cancers. So, similarly, all of these genes can give rise to numerous cancers. Here, the HER2 new gene that can give rise to breast cancer, or which one? RAS is a very broad cancer. It can give rise to multiple different cancers. But the ultimate idea is that all of these genes they promote growth, and what the cancer does is it hijacks these genes and it causes more growth signals to be produced, because of which it will be able to grow more rapidly. Additionally, the cancer wants to decrease the levels of anti-growth hormone or anti-growth signals so that these growth signals they can work even better and the cancer can replicate further. Now, cancer also wants to prevent its own death. That means the cancer also wants to prevent apoptosis. And to prevent apoptosis, it has multiple different things that it can do. The most common thing that cancer does is that it hides its own MHC class. So that means cancer cells that do not express MHC class. Now, if the cancer does not express the MHC class, which cells is it protected against? So what do you guys think? If the cancer does not have a MHC molecule, then which cells is it protected from?
Is it T cells?
Yeah, it is T cells. So the cytotoxic T cells, it is protected from them. But on the other hand, the cancer is more vulnerable to the NK cells. And then what it does to like protect itself against the NK cells is it like releases more cytokines which can kind of neutralize the NK cells. And that is how the cancer kind of masks its identity. So the next one is limitless replication potential. Now here there is one very important enzyme and this is called telomerase. Now what telomerase does is it is basically a end point of at the end of the chromosome. There's a loop of unused like DNA which kind of shortens every time there is a replication. And this is to protect the DNA from being damaged during replication. Over the span of a cell's life, this telosome or these telomerase molecules, they'll start degrading. However, cancer has a specific enzyme which is telomerase and it will produce these telomers. So the cancer can, in theory, ultimately keep on growing. Then we have sustained angiogenesis. That means the cancer, because it's growing so rapidly, it requires a lot of energy. And all of this energy will come from blood. So what the cancer wants to do is form more blood vessels around itself. And the more blood vessels it forms around itself, the better it will be able to grow. So everything that we've talked, from increasing the growth signals to inhibiting the anti-growth signals to evading apoptosis, limitless replication, sustained angiogenesis, all of these things, they're all only there for allowing the cancer to grow even more. Now, next we have Wahlberg effect. Now, Wahlberg effect basically means that glucose in normal cells it goes through glycolysis, Krebs cycle, and then ultimately through the etc. In cancer cells, though, what happens is there is a decrease in aerobic respiration, and instead the cancer focuses more on anaerobic respiration. So, why do you think the cancer is focusing more on anaerobic respiration compared to aerobic respiration?
I'm not sure.
Because, for an aerobic respiration, basically you'll have to go through glycolysis, then you'll have to go through the Krebs cycle. After that, you'll have to go through the etc. So it's taking a lot of time.
Constipation
Yes,
waste
if
time.
if the cancer doesn't care about the body, then what it can do is it can use up all the glucose just as quickly as possible, convert everything into lactate or ferment everything, get as much energy as possible, and then not care about anything because the cancer isn't normal functional tissue. It doesn't follow the body's rules, I would say. So that's why it just completely goes anaerobic because that's faster. And then cancer has like ultimate, like unlimited blood supply because of the sustained angiogenesis. And then the next one is again evasion of the immune system, what we talked about. Now here, these two, they're pretty similar, where cancer, it wants to grow, and in addition to growing, it also wants to spread. So first it invades the tissue, that means it gets inside a tissue, tries to invade into the blood vessels, and then ultimately it metastasizes and goes to other locations to propagate its growth. So guys, any questions until this point?
That's good.
So the next topic that we have is basically the immune checkpoints. Now these immune checkpoints, they're basically how does the, let's say, oh yeah, it's immune checkpoint interaction. Okay, yeah. So this immune checkpoints is basically how does T cell get activated and what's the interaction between T cells and the tumor cells. So normally the MHC class 1 would be active and the tumor cell will produce certain proteins which would be presented on the MHC class 1. And if this is a foreign protein that the T cell does not recognize, it will activate the T cell and cause it to kill the tumor cell. Once this happens, and this is very similar to what we did in immunology, that once a tumor cell is killed, then all of those antigens from the tumor cell would be sent to the antigen presenting cells, and then that would ultimately lead to a cascade where all of the different immune cells are activated. Here, wait. Oh, yeah. So there are a couple of additional things that the cancer does. Now, the most important one here is the PD1, PDL1 interaction. And I think we've done a question on this, where the cancer, in addition to the MHC class 1 molecule, it also has a PDL1 molecule. And this PDL1 molecule basically is the same molecule which is responsible for inactivating T cells in the thymus. Now, if a T cell does, let's say, come to destroy a cancer cell, and you have a interaction of the PD1, PDL1 ligands, then that basically inactivates the T cell, and it inactivates the T cell in such a way where the T cell would not be able to be activated again. It's in complete energy, and that is why this is dangerous. Now, there are some medications that we've developed over time to combat this. So we have a medication which blocks the PD1 receptor. We have medications which block the PDL1 receptor. We also have other medications which block, let's say, CTLA4, which is again responsible for inhibiting T cell function. So the ultimate goal in cancer treatment is to, I would say, keep our T cells active for as long as possible and to have them active at the highest level of activity as possible so that they can go ahead and kill the tumor cells. Now, this chart is also very important that the most common cancer in males is the prostate cancer, and in females, it's breast cancer, and that is followed by lung cancer in both genders and then colorectal cancer. However, this is not the so this is basically cancers by incidence, so how frequently they happen, and all of these pale in compared to like skin cancer, which is the most common type of cancer. And then after that, we have the mortality. So how likely is a cancer? How likely is it for a cancer to kill someone? And that is lung cancers as the highest level of mortality. After that, we have prostate cancer, and then we have colorectal cancer. Here, one more thing is that cancer is actually the second leading cause of death. Overall, cancer is the second leading cause of death, and heart disease, so cardiovascular disease, is the first, and then third would be cerebrovascular incidents. So this, I would say, very important to remember, and you should remember. If not for, you can forget the children kind of aspect of it, but you do need to remember these two. Now, for metastasis. Now, for metastasis, right? Let's kind of make an assumption. Which type of organs would have the highest rate of metastasis?
Anything that goes with travel around body organs.
Yes, anything which gets a lot of blood, anything which gets a lot of lymph will have a very high risk of metastasis. And that is why, liver has such a high risk of metastasis. The lungs, the brain have such a high risk of metastasis because a lot of blood goes to these areas. Even kidneys have a high risk of metastasis, though doesn't really happen that often, but they can also have a lot of metastasis. Bones also have a lot of metastasis. However, the reason why bones have a lot of metastasis from the prostate and the breast cancers is because of proximity. The lumbar spine is very close to the prostate, and if you have a prostate cancer, you're very certain that this cancer will ultimately travel to the bone, the lumbar spine, and that is why bone is mentioned here. Similar for breast cancer. For the breast cancer, it can also travel to the spinal column, and that is why like we also have bone cancer, which is mentioned. Now, you need to remember what type of lesions will you find when you have bone metastasis. So for prostate, we know that it will usually be a blastic lesion. That means it will be more osteoblasts within the cancer or the bone where the cancer has embedded. And on the other hand, if you have a breast cancer, then you'll have both osteoblasts and osteoclasts together in the lesion. One more thing is that whenever you have a cancer which has metastasized, and this is actually something I want to ask you guys. If you have a cancer which has metastasized, do you think you'll see one lesion or multiple lesions?
Multiple lesions.
And why multiple lesions?
Because it is spreading like from one side of the
Yeah, because it's spreading. And an even more nuanced reason would be that the entire cancer doesn't spread. It's small particles or small clumps of cells of the cancer which kind of break apart and they spread. And that is why you have multiple lesions. So if you see multiple small lesions, let's say affecting the brain, then you can say that this is a brain metastasis. Let's say there is multiple lesions which are affecting the lungs, both the lungs. Then you can say that this is a lung cancer. In the lung, it will usually be described as cannonball lesions. So it'll have multiple small round lesions in the lungs. And then next we have the most dreaded portion of oncology, where you need to remember all of these genes and both of these kind of tables. So you need to remember what the gene is and which type of cancer it causes. Now you don't need to remember all of them, but it would be good if you could remember a majority of them. So here you have the MYC genes, you have KRAS for colon and like GI cancers, you have BCL for like leukemia or lymphomas, you have HER2 which is for breast cancer, as I said. You also have BCR-ABL for CML and multiple different cancers. So you do need to remember this entire table and also this table. You need to remember all of these cancers. Now, you don't need to remember what exactly all of these cancers are, because when we study the different systems, you'll automatically kind of come to understand what these cancers are. Let's say NF1, NF2. But you do need to remember what the gene is, what it produces, and then what type of cancer it causes. Yeah. And then one more for the carcinogens. You also need to remember these carcinogens. So I can say one thing that for these carcinogens, the tumor suppressor genes, and then also the oncogenes, I will try to add as many like questions in the group as possible, so that like the WhatsApp polls that I used to add. So I can add those, so you can mark which of them would be the correct answer. So we constantly have a review of these oncogenes. But yeah, you do need to memorize these. Yeah. Okay. Finally, for today, we'll end at this topic, which is field cancerization. Now, field cancerization is when you have an entire area. Usually, cancer it develops from one cell or a small group of cells, and that can lead to like ultimately it grows into a bigger cancer. But sometimes when you have like exposure to a very strong carcinogen, you can develop a very large field where like all cells have turned into cancer. Now, this is bad because like one small cell, if it turns into cancer, it's already a problem. So if you have an entire group of cells which are turning into cancer, then that becomes very problematic because it can grow at an even faster rate. And that is field cancerization. If an entire group or like a large area of cells is converting into cancer. Now, here they say pre-malignant cells because our entire goal in treating cancer is not to let cancer develop. We want to find the cancer before it develops. And that's why they say pre-malignant cells. But if given some time, they will obviously develop into malignant cells. That's the idea of field cancerization. So, guys, any questions about the topics that we did until now? Yeah, Devanshi.
Um, in the cancers we should discover from health which spreads through blood, so only those cancers which spread from blood rest are one transmission because transmission is like it's spreading from
Yeah, so usually, all cancers they transmit or like kind of go through the lymphatic system. They go to the lymph node, and then from the lymph node, it can spread. There are only like four cancers which can directly go to blood. Ultimately, all cancers will go to blood. So they'll first go to the lymphatic system. From the lymphatic system, either they go to a bigger lymphatic organ or they can go into blood.
okay, so can you please name those four again?
Oh, yeah, I can send a message in the group with these four.
Okay.
So you have renal cell cancer, then you have hepatocellular cancer, then you have malignant melanoma, and then you'll have to check this one, but I believe it is follicular cancer of thyroid.
Okay, let me check.
Okay. Uh, yeah. So
Thank you so much, Dr.
no problem. And then Kizzy also had a question, or was it? Oh yeah, Kizzy had a question. So basically, it was what does PD1 and PDL1 do? Basically, which let me open the image. Oh, okay, no problem. So, when we were studying the immune system, do you remember in the thymus we said that we want to inactivate cells? Both, we had two selections. We had a positive selection and we had a negative selection. Now, these selections, they're basically, you're either like completely killing of a cell or you're completely stopping the cell. So it's either apoptosis or energy. Energy basically means that the cell is inactive now. It cannot be restarted again. One second. Yeah. So basically, it means that the like cell it cannot be restarted again for energy. Now, PD1 and PDL1, basically this interaction is the one which causes the cancer to be inactive. Sorry, not the cancer, the T cells to be inactive. So cancer, it ultimately developed a molecule, the PDL1, which mimics what happens in the thymus. That if you have the interaction between the PD1 and the PDL1, then the T cell becomes immobile. It cannot work anymore. And this is good for the cancer because now the cancer doesn't have to worry about the T cells. And that is how this happens. Okay. So, guys, any more questions?
We are good for this.